Okay, we're going to continue our discussion of electrical potentials and how we establish an action potential in a nerve cell. So in order for this woman to move her foot, she needs to transmit an impulse from a thought in her brain down the spinal cord to the nerve serving the foot. This can happen very quickly as an impulse travels down myelinated axons. So today we're going to examine exactly how that process occurs in neurons and how it's transmitted along the axon in myelinated axons. To begin, you want to look at your PowerPoint notes. We're looking at specifically slide 11-9, and we'll follow along with this tutorial from there. So recall that nerves are bundles of axons that are myelinated, and these axons are very long. They can begin from the base of the spinal cord all the way down to the muscles serving her foot in this example of the woman tapping her foot. So there is a long axon carrying information, and the faster that information is needed to be conducted, the more likely we're to see myelinated axons. Most of the nerves serving the muscles are myelinated axons. So this is a picture of an axon, and we can see that there is more positive charge on the outside of the axon, more negative charge on the inside, and that those charges reverse themselves when an action potential travels down the axon. So we're going to just look at how these charges are distributed across the axon. So again, you can see how this impulse here is what's the bar traveling down the axon is simply an opening and closing of ion channels which cause ions to flow across the membrane and reverse the charge. So we're going to look at how this difference in charge is established across the membrane. This being the inside of the axon and then the positive on the outside. So recall that ions have specific charges. We have a number of ions that we find distributed across our cell membranes. The two ions we're going to focus on are potassium and sodium. Notice they're both positively charged ions. So if we look at the plasma membrane of an axon on a neuron, we'll see two different types of channels. We see sodium channels that only conduct sodium ions, and we'll see potassium channels that only conduct potassium. So here we see a sodium ion traveling through a sodium channel and a potassium ion through a potassium channel, and they follow their concentration gradients. Recall what establishes the concentration gradient is the sodium-potassium pump. Remember, it brings 3 sodium out and 2 potassium in. As a result, we're always going to find more sodium outside the cell and more potassium inside the cell because of this pump. So potassium always naturally diffuses out while sodium naturally diffuses in because of the concentration gradients created by the sodium-potassium pump. So the sodium-potassium pump is a key player in establishing this charge. So because of that, because we are losing potassium, which is a positively charged membrane, or positively charged ion, that results in the inside of the cell being slightly negative compared to the outside of the cell. We have lots of sodium ions on the outside of the cell because of the sodium-potassium pump, and we have constant flow of potassium outside, along with some negatively charged proteins on the inside, again leaving the inside of the cell negative and the outside of the cell positive. So if we distribute these ions, we would see that we're going to find more sodium on the outside because of the pump, pumping 3 out, and we're going to find more potassium on the inside because of the sodium-potassium pump pumping it in. So if we just distributed those ions, we see that definitely there is a difference because of that pump. So we see more potassium inside, more sodium outside because of the sodium-potassium pump. So can you see why we have a more positive charge on the outside? Because of these many positively charged ions in combination with the flow of potassium out as a result of the concentration gradients. When we have more potassium in compared to out, potassium wants to flow out of the cell. So if we want to make the inside of this cell more positive, we call that a depolarization, and that's what starts an electrical impulse in a cell. So to make this cell more positive, we would have to bring positive charge into one of these channels. Well, recall that potassium channels are always carrying potassium out because that's the direction potassium travels following its concentration gradient. Sodium, you can see the arrows are pointing in because sodium will always diffuse in because of its concentration gradient. It's again established by the sodium-potassium pump. So if we want to have a depolarization, which means we want to take the inside of the cell and make it more positive, we need to bring more positive ions in. And the only way we can accomplish that is through bringing sodium in as a result of its diffusing down its concentration gradient. So I'm going to close this channel. I'm going to leave this one open. Whoop. And we'll see how sodium travels into the cell and how that's going to change the concentration gradient of this cell in the positive direction. Because right now we have a charge of -70, which means it's -70 millivolts more negative compared to on the inside compared to the outside. So with this open channel, we're going to find that sodium flows into the cell. And notice the rise in the graph as the inside becomes more positive, and now the outside is more negative. Again, the sodium-potassium pump is going to reestablish those gradients. Now, if we want to make the inside of the cell negative again, relative to the outside, we would have to move a different ion. So again, if we want this to be negative, that means we need to move potassium out of the cell and close this sodium channel. So I'm going to close this channel, and I'm going to keep this open, and that will cause potassium to flow out of the cell. Potassium is a positively charged ion, so as it flows out, it will create negativity on the inside, because if we lose positive charge, that creates a negative charge inside the cell. And we'll watch that happen. Potassium flows out of the cell. We see a negative drop here, return to negativity, and it's negative on the inside of the cell once again. Now, what's going to establish concentration gradients again is the sodium-potassium pump. So notice this little spot here is the result of the sodium-potassium pump reestablishing those ions on the correct side of the membrane, cycling 3 sodium out, 2 potassium in, as long as there's ATP available. So this process is the result of these channels is the action potential. So these are voltage-gated channels that only open at certain voltages. And in this case, potassium channels, voltage-gated potassium channels, only open at positive membrane potentials. In this case, plus 30 is when the potassium channels would open, and sodium channels only open at threshold. Voltage-gated sodium channels only open at threshold. And threshold is a specific millivolt that is specific to the particular cell. And in your textbook, the threshold is shown as positive 30 millivolts. I'm sorry, not positive 30. I'm sorry, threshold is at different textbook, use different numbers. Let's see if I can find the number for this book. Threshold. Look at your textbook. In chapter 11 is the chapter that we're in. And specifically the action potential is really well described on page 402 and 403. And threshold in this case is -55. So when this negativity, when we have enough sodium flowing in to reach -55, that is going to cause all the sodium channels to open in that portion of the membrane, and then sodium will come rushing into the cell. So we see this steep climb here once we get to -55, which we call threshold. So how do we reach threshold to get these voltage channels to open? We need a little bit of positivity inside the cell, going from -70 to -55, and how we accomplish that is through sensory receptors. So sensory receptors are specialized parts of neurons that have sodium channels that will open in response to a stimulus. That in the case of neurons in the eye, it would be light that would stimulate some sodium channels to open. In the case of ears, it would be sound vibrations that would cause sodium channels to open. So the important concept is that we are constantly stimulating sensory receptors to open up sodium channels to reach threshold. So let's go back to our PowerPoint here and look at the action potential graph. So when I reach threshold, here's an example right here. This is the best one that shows it. So if I'm here sitting here at -70 in a neuron, and that again is based on potassium leaving the cell, sodium entering, but more sodium being dumped out of the cell and potassium leaving by diffusion, that we get this negative potential inside the cell of -70. So I have enough stimuli, enough light or enough sound or enough pressure, temperature, touch to open up some specialized sodium channels on the sensory receptors. If I get enough stimulus, we call that a local potential. If I get enough sodium flowing into the cell to reach -55, then voltage-gated sodium channels all rush open on this portion of the membrane, causing an action potential. And this occurs on the axon hillock. And the axon hillock, if we go back and look at the structure of a axon, is right here. So out here in the dendrites, we have specialized sensory receptors that open up sodium, that have special sodium channels that open in response to a stimulus. And if we get enough sodium flowing into the cell, that positive charge will be enough to reach threshold, and at the axon hillock is where we see a concentration of these voltage-gated sodium channels, and that will stimulate the -50, or that will result in a -55 millivolt threshold and cause all the voltage-gated channels to open in that area, and then that impulse will travel from point to point on the membrane, again from node to node to node. So we see a concentration of sodium and potassium voltage-gated channels at the nodes of RANVA. So it's really important that we understand, when we look at this graph, exactly what's happening on the membrane. So here we have the sodium-potassium pump. We have leaky channels for potassium and sodium just doing their job. Then we have some type of stimulus occurring out on the dendrites, opening up some of the sodium channels that are specialized for that stimulus, allowing positive sodium to enter the cell. Once we reach -55, then that triggers voltage-gated sodium channels at the axon hillock to cause an action potential to develop down the membrane. And this rapid rise due to sodium rushing into the cell through voltage-gated sodium channels is called depolarization. Once it reaches positive 20 millivolts inside that cell, those voltage-gated sodium channels close, voltage-gated potassium channels open, and potassium goes rushing out of the cell with its concentration gradient. We call that a repolarization. As positive potassium leaves the cell, that makes it more negative again, so we see this drop in the graph all the way down past -90, because sodium channels are slow to close. We get a little extra sodium leaking out. And then again, the sodium-potassium pump cleans things up. This is called the after potential, and that brings us back up to resting, which is -70. So it's very important that you be able to label this graph with what channels are open and what ion is moving back and forth across the membrane. So you might need to replay this video to listen to my explanation again to determine what channels are open and what ion is moving and in what direction. And that concludes part 2 of our discussion, and we'll wrap up the rest of the nervous system when we meet together in class.